Welcome to Cell Guidance System webinar. So today we're really excited about the talk of Dr. Petr Baranov, which is going to talk about improving neuron differentiation outcome with a slow release growth factor system. So this slow release growth factor system is one of Cell Guidance System product. So I'm just going to do a really brief introduction about this technology, and then I will let Dr. Petr talking about his research and how did he use our product. So really briefly, as I said, so this slow release growth factor is named PODS. For polyhedrin delivery system. So the polyhedrin protein is a protein that will self-assemble into a cubic form, as you can see on the picture. And our technology co-expresses this protein with the growth factor. And this enables the protection of the growth factor into, as we call, a protein shell. And then it can protect and preserve the function of the growth factor. There is a slow release over time, so it's from 2 weeks to 12 weeks, depending on your needs, on your protocol, on your cell type as well. And you can also modify this release by combining this PODS crystal with different scaffolds. If you're working, for example, with hydrogel or with matrix protein, they can be mixed together. PODS crystal can also be used to create gradients. So you just have to spot them on a specific place, and then they will create a gradient over time exactly on the at the spot when you pipette them. So it's a really interesting technology that Petr is going to talk today. If you're interested in learning more about it, you can contact us by going to our website or by contacting me directly on my email address that you can see on the slide. And so now we are really happy to introduce you to Dr. Petr Baranov. He is from the Institute of Massachusetts Eyes and Ear at the Harvard Medical School, and he's going to talk about improving neural differentiation outcome with our slow release growth factor.
Hey, thank you, Julia, for the introduction, and thanks everyone for joining us today. I'll try to keep it brief and not waste too much of your time. And in the next 15 to 20 minutes, I'm going to tell you about the work that we have been doing in the lab trying to replace retinal ganglion cells. Before I proceed, I'd like to acknowledge all the hard work done by lab members and especially Julia Oswald, who executed most of the experiments I'm going to cover today. Where do we start? For those of you who is not familiar with the retina, you can really call it the approachable part of the brain. It makes a very valuable model to explore neuron development and transplantation for us. And one of the advantages, it is a very well-defined set of neuronal classes and subtypes. There are photoreceptors, bipolar cells, horizontal and amacrine cells, and of course, retinal ganglion cells, the type we're going to talk about today. These neurons reside in the innermost of the retina, close to the vitreous, and they have very long axons that extend all the way through the optic nerve and optic chiasm into the brain. Unfortunately, the mammalian retina has very little and almost non-clinically relevant regenerative capacity, and that's why we do what we do, trying to replace retinal ganglion cells lost in diseases like glaucoma or traumatic optic neuropathy. And the way we approach it is really by trying to recapitulate RGC development first in vitro from renewable cell source and then in vivo. Where do we get retinal neurons? The answer would be obvious for most of you, of course, to make them from stem cells. And since 2011, the retinal differentiation became much easier and way more reproducible because of the amazing work done by a group of late Ishiki Sasai. Briefly, they have shown that it's possible to truly recapitulate retinal development in three-dimensional organoids. And for those of you not familiar with this paper, I highly recommend you to look it up. What is unique about this protocol, it is very well conserved in mammals, and since 2011 it has been reproduced multiple number of times with mouse, pig, non-human primate, and of course human pluripotent stem cells. How does it work in our hands? We start with fibroblasts from transgenic reporter mouse strains. In this case, the data I'm going to show you today would be generated with Thy1 GFP cells. Thy1 is a marker for neurons, and in retina, it is specific to retinal ganglion cells. In developing retina, it's specific to retinal ganglion cells. So we start with the fibroblasts. We induce pluripotency with classic Yamanaka factors, and after expansion and characterization of pluripotent stem cell line, we dissociate them and put together to form an embryo body. In several days, as you can appreciate from these bright field images on the left, we observe neuroactive formation, followed by optic vesicle induction and optic cup formation, and we can mature these organoids for several weeks in order to get individual retinal neurons like ganglion cells or photoreceptors. As you can see from this image, if we grow them longer, these neurons start to acquire proper morphology and send their projections all the way across the organoid. When it comes to individual cell types and numbers, for ganglion cells, we get the range of about 1.5 to 7% of cells, and we also can pull them out from the organoid by sorting for CD90, which is a surface marker, or another marker, L1CAM. Another thing you need to know about RGCs to truly appreciate the utility of this system is that retinal ganglion cell is a very heterogeneous and diverse population. In mouse retina, there are more than 35 subtypes that can be distinguished based on their structure, function, and molecular profile. There are on-off alpha RGCs, on direction-sensitive ganglion cells, alpha RGCs. There are intrinsically photosensitive RGCs. The two types that we are most interested are intrinsically photosensitive RGCs. So these would be the cells that express melanopsin and can sense light without photoreceptors, and also alpha RGCs. These are the cells that are, I would say, most robust and show the most regenerative capacity when it comes to axon damage and axon regeneration studies. So when we stain our organoids, stem cell-derived or retinal organoids for individual retinal ganglion cell markers, we do observe a very nice pattern. And I should add that flow cytometry data shows that these markers do not overlap, which confirms that these are not some chimeric cells that just express everything, but rather individually mature cell types. So why are we talking about growth factors and slow-release delivery system? The reason for that is that once these ganglion cells are born in the organoids, they are on the path to death. If you do nothing, then in about 10-15 days, most of them will disappear from the cultures. And we try to create better maintenance conditions for them. And one of the approaches would be to supplement them with no neuroprotective factors. The direct addition of growth factors was not as effective, so we tested several slow-release systems, and POD was one of them. And one of the experiments I'm going to show you is that when we differentiated the retinal organoids with our protocol, and at day 20, we supplemented the culture with BDNF and GDNF. The reason to select these two factors over the others is because they have shown very high potency for photoreceptor and ganglion cell rescue both in vitro and in vivo in various retinal degeneration models. And then 10 days after, we collected the organoids, dissociated them, and performed a basic cell count and retinal ganglion cell count using flow cytometry analysis. So what we can appreciate from the graph on the left, that we did observe significant rescue over the course of 10-day treatment. And what is also important here, that effect is dose dependent. So when we use low concentration of pods, we didn't observe the effect in comparison to our control group, which were antipods in this case. And this is something you have to keep in mind when you try to estimate the most effective concentration for your particular cell type, your particular system, and I think a lot of things are important when you consider the growth factor to work with as well. When we look into the individual cell types, and these are the cell types I mentioned already, osteopontin positive alpha RGCs and melanopsin positive photosensitive retinal ganglion cells, we observed almost twofold increase in their numbers compared to the controls. So what do we do with these cells? And now we have a cell type we can work with. If we want, we can dissociate them and place them on a surface for functional analysis. But our overall goal is really transplant them and let them grow in the eye, in the degenerated eye. So we try to use the slow-release delivery system to create conditions in vivo to support donor cell development and integration. And before executing this experiment, we've done several sets of transplants. And one of the things we discovered is that donor RGCs do integrate better in damaged retina. We also have shown that they integrate better in the developing retina, which giving us a clue that there may be something about the environment that affects their integration. So for this experiment, we isolated RGCs also at day 21 using magnetic microbeads. We formulated them with pods and then performed basic intravitreal injection of about 20,000 cells. And 2 weeks after that injection, collected the retinas, performed the dissection and staining for further histological analysis. As I said in the beginning of the talk, that we do believe the eye to be an approachable part of the brain, and this is the reason for that, because all the optics is there, and you can really trace individual donor cells if they have any kind of reporter system in them. You can see their processes, you can see their migration, you can even do a basic cell count. And of course, when you extract their retina out of their eye and perform histological analysis, you can look into this at much higher resolution. So what we have here are 2 examples of host retinas 2 weeks after transplantation with donor cells in green. And on the left side of the screen was the retina which received only donor cells. And on the right side, the retina that received both donor cells and growth factors. I don't think we need to do any statistics to show that there was a striking difference. And to us, it was really impressive, the outcome of the study. While the overall transplant success rate, which is a number of animals that have detectable number of donor cells, the overall transplant success rate remained almost the same. The area for transplantation, then the number of donor cells, and the process outgrowth was really striking. When we look with high-res magnification, we saw that the donor cells in the group that also received growth factors, they responded as they responded in developing retina and sent their axons all across the retina, adjacent to the host axons. And what is more important for us when it comes to retinal regeneration and cell replacement therapy is that these axons were really capable of going into the optic nerve head, something that remained a challenge for a long time. But here I would bring up one more shot of the transplant to show that axons can indeed go into the optic nerve head and follow the host axon boundaries for excessive length outside of the retina. Another parameter that we use to assess the maturation of structural maturation of donor cells in after transplantation is the neural outgrowth and the overall survival. So you can also appreciate on the left in the images showing that cells supplemented with growth factors exhibited much higher rate of neural outgrowth. I think for the sake of time, I'm going to stop here and would be happy to take any questions. Thank you.
Thank you very much, Petr, for this really interesting talk. So now if anybody has questions, Petr will be happy to answer any of these. Hi, Petr.
Good afternoon. Hi, everyone. I have a question from Sanj asking how applicable are pods for other systems. In our hands, we have not tried them for other cell types, but I think what we know about the universal approach for neuroprotection with growth factors, I think that should work as well for other neuronal types. Okay, question from Leo about have we assessed the functional capacity of neurons we're making, either in vitro or in vivo? The short answer is yes. We're interested in retinal ganglion cells, and we tested their ability to respond to glutamate. And we also have performed some basic functional assays in vivo using electroretinography to measure retinal function, and also using agmatine to measure individual cell function. We don't have any data showing that pods or growth factors that we live with pods affect individual cell function by now. I have a question from Carolina. That's a technical question. If pods remain attached to the culture plates, I mean, looking at the media, they probably do, but I think Julia will be able to address the question better when it comes to what is the percent of loss of these crystals when you do media change. You want to address that, Julia?
Yeah, sure. So we did it in-house, we tested it. So yes, the pods crystal will adhere to plastic. So when you do the media changes, the percentage of loss is really small, is less than 1%. So they stick really hardly to the plastic.
Thank you. I have a question from John Fuller about the percentage of retinal ganglion cells in eye cups that we generate, if it matches the in vivo retina development. So if we look at later stages of differentiation, it does match the percentage of retinal ganglion cells in primary retina. At earlier stages of differentiation, it's actually higher than percent of retinal ganglion cells in primary retina. So if in primary retina is about 1%, the numbers we see with earlier stages can reach 5-7% of retinal ganglion cells per organoid.
So maybe we will wait a few minutes to see if there is any other questions.
All right. A related question for potential clinical or therapeutic application and what we do with the retinal ganglion cell replacement. And Michael asked what would be the biggest hurdle. And this is something we really work on in the lab. And our target is glaucoma. And the major challenges that we have right now is a proper structural integration into the retina. We want to make sure that axons would enter the optic nerve head and go all the way to the brain. And then something we cannot address right now is how specific would be the connections they make in the brain. Do they go to visual cortex? Do they go other places? And if synapses they form would be refined with time. But we hope we'll be able to resolve it in animal experiments. And one question with Sanj for the future experiments. In terms of this project and looking at the neuroprotective action for in vitro experiments, I think it would be interesting to see if we can modulate the function. And something that we are interested in is maturation of neurons. If we can improve their function, the electrophysiology with prolonged cultures where we can modulate microenvironment using growth factors. And for in vivo experiments, I think it would be the same. So the major focus is on function, because really achieving functional integration of neurons into the retina is our target.
Thank you, Petr. It was really interesting. It's nice to see how our project can actually be used in an application. I think it's better to understand how to use them, through your research now. So thank you, Petr, and thank you everyone for attending this webinar. I hope you enjoyed it, and we hopefully will do a webinar on post-crystal again, but for another application. So thank you very much for everyone, and have a nice day, evening, afternoon.
Thank you.
Bye.